What if one of the deadliest events in human history did not end when the bodies were buried? What if the Black Death changed which people survived, which families continued, and which versions of the human immune system were passed down through generations? Some scientists believe traces of that catastrophe may still be present today, not as disease but as DNA variations shaping immune response. A past genetic advantage may carry a modern cost, and the claim remains one of ancient DNA's fiercest debates. Between the late 1340s and early 1350s, a devastating pandemic swept across Europe, North Africa, and parts of Asia. The disease was caused by the bacterium Yersinia pestis. In affected regions, the death toll was catastrophic. Entire households disappeared. Communities lost a huge proportion of their population. Social structures, economies, and daily life were transformed. For centuries, historians studied the Black Death through burial records, chronicles, and mass graves. Today, scientists can investigate it in another way. They extract tiny fragments of DNA from the teeth and bones of people who lived before, during, and after the pandemic. Those fragments provide a partial record of biological inheritance. By comparing genetic variants across time, researchers can ask an extraordinary question. Did the Black Death kill randomly, or were some people genetically better equipped to survive? If immune system variants helped carriers survive, did those variants become more common later? A major 2022 study identified a region near the immune gene ERAP2 as the strongest candidate for selection. The finding generated dramatic headlines. The plague had left a scar on the human genome. Then other geneticists re-examined the evidence and challenged whether the evolutionary change had been demonstrated. The Black Death may have shaped immunity, and ERAP2 may be involved, but science has not delivered a final verdict. Evolution is often imagined as a process occurring over vast stretches of time. Useful traits become more common across thousands of generations. But under extreme conditions, natural selection can act much more quickly. Imagine a population containing several versions of an immune-related gene. One version might let immune cells detect or control a pathogen more effectively. Another might produce a weaker, slower, or simply different response. Under normal circumstances, the difference might be small. But during a pandemic, killing an enormous proportion of the population, even a modest survival advantage could matter. People carrying the more protective version would be more likely to survive, have children, and pass it forward. After the pandemic, its frequency could rise. This is the basic evolutionary hypothesis behind the Black Death research. But testing it is extremely difficult. Ancient DNA does not arrive as a clean genome. After centuries underground, heat, water, microorganisms, and chemical reactions damage it further. Human remains may also contain DNA from soil organisms or from people who handled them. Researchers therefore need carefully dated samples and reliable archaeological context. They also need enough individuals from different periods to detect meaningful patterns. Even then, a change in gene frequency does not automatically prove natural selection. Populations migrate. Groups replace or mix with one another. Families may be overrepresented in burial sites. Random genetic drift can alter frequencies, especially in small samples. Technical differences in DNA capture and analysis can also create apparent changes. Scientists are reconstructing a continent-wide evolutionary event from a limited and imperfect archive. It is like trying to understand an entire storm, using only a few scattered drops of rain. For the influential 2022 study, researchers examined ancient human remains associated with London and Denmark. The London material came from people buried in and around the medieval city. The Danish material provided a second population for comparison. The samples represented individuals who lived before, during, and after the Black Death. The researchers focused heavily on immune-related regions of the genome,
looking for variants whose frequencies appeared to change across the pandemic period.
Their analysis highlighted hundreds of differentiated variants in the London dataset, with a smaller number of candidates also appearing in the Danish material. The most prominent signal was found near ERAP2. ERAP2 is not a plague gene. It is a human gene involved in immune function. Inside our cells, proteins are constantly broken into smaller fragments called peptides. Some of those fragments are displayed on the cell surface by molecules known as HLA class 1.
This display system helps immune cells inspect what is happening inside other cells.
ERAP2 helps trim certain peptides to appropriate lengths before they are presented. Think of it as part of a molecular editing system. The cell breaks proteins into fragments. ERAP2 helps shape some of those fragments. The immune system then examines the finished pieces for signs of infection or abnormal activity. Common genetic variation affects whether people produce a normal, full-length ERAP2 protein. Some versions of the region are associated with greater ERAP2 expression, while others can produce transcripts that are degraded before a functional full-length protein is made.
The 2022 researchers argued that one ERAP2-linked variant rose substantially around the Black Death.
If correct, that would represent remarkably strong natural selection over a short historical period.
But statistical frequency was not their only evidence. They also tested whether the variant changed immune responses to Yersinia pestis.
The team exposed immune cells from people with different ERAP2 genotypes to Yersinia pestis in laboratory experiments. They reported differences in cytokine responses and in the ability of macrophages to control the bacteria. Macrophages are immune cells that engulf microbes and damaged material. They are among the body's early responders to infection.
According to the study, cells with the proposed protective ERAP2 configuration were better able to restrict intracellular Yersinia pestis.
This functional experiment mattered. Ancient DNA studies often identify a statistical pattern, but cannot demonstrate what the genetic variant actually does. Here, the researchers had an apparent historical frequency change and a biological mechanism that could plausibly affect infection. Their interpretation was dramatic. People carrying 2 copies of the advantageous ERAP2-associated variant may have had a significantly greater chance of surviving the Black Death than people carrying the alternative version. But this should not be understood as a precise survival prediction for any individual. A medieval person's fate would also have depended on age and health, nutrition, exposure and living conditions, the form of plague involved, and many other genetic and environmental factors. ERAP2, even under the original interpretation, would have been one factor, not a magical shield. And there was another complication. A more active or responsive immune system is not universally beneficial. The same machinery that attacks infection can also contribute to harmful inflammation. A genetic trait can be helpful in one environment and disadvantageous in another. The immune system must constantly balance 2 dangers. Respond too weakly and infection may spread. Respond too aggressively and the body may damage its own tissues. Genes involved in antigen processing and inflammation can therefore produce trade-offs.
The 2022 study linked the proposed plague-protective ERAP2 configuration with modern inflammatory or autoimmune conditions, including Crohn's disease.
Later research examined ERAP2-related variation and found opposing associations between severe respiratory infection and Crohn's disease. Reduced ERAP2 expression was linked to slightly higher odds of severe respiratory infection, but lower odds of Crohn's disease in those datasets. Researchers interpreted this as possible balancing selection. Different forms of a gene may remain because each carries advantages and disadvantages. This does not mean the Black Death directly causes Crohn's disease today. Crohn's disease is complex. Genetics, immune activity, microbes, and environmental factors all contribute. No single medieval event explains why one modern person develops it. Nor does carrying a particular ERAP2 variant determine that someone will become ill. Genetic associations describe shifts in probability across populations. They are not diagnoses. Still, the possibility is profound. An immune response favored in a world of frequent lethal infection might operate differently in a modern environment shaped by sanitation and antibiotics. Vaccination, altered diets, and longer lifespans change the environment too. Evolution does not design bodies for permanent health. It favors traits that improve reproductive success within a particular environment. If a variant helped people survive repeated epidemics long enough to have children, evolution could favor it even if it caused problems later or elsewhere. That is the trade-off hypothesis at the heart of this story. But showing modern ERAP2 effects is different from showing the Black Death shaped its frequency. The first has substantial genetic and functional evidence. The second remains disputed. After the original study appeared, other population geneticists examined its assumptions. Their criticism focused on whether the apparent changes were strong and reliable enough to prove natural selection during the Black Death. In a formal challenge published in Nature in 2025, researchers argued that the evidence was insufficient. They raised several concerns. First, the ancient sample sizes were limited. Second, the before and after groups covered broad and sometimes overlapping date ranges. Third, immune and neutral regions may have been captured and measured differently. Those technical differences could create apparent genetic changes. Most importantly, critics could not confirm a significant change in the frequency of the headline ERAP2 variant. When critics examined additional ancient genomes spanning roughly 2,000 years, they found no clear rise matching the dramatic evolutionary shift originally proposed. That did not prove the Black Death had no evolutionary effect. It challenged whether this dataset could establish one. A separate ancient DNA study of medieval Cambridge complicated the picture further. It found no clear evidence that specific variants let some people survive plague. That added doubt to the idea of a simple, universal plague survival gene. The original research team responded. They argued that critics placed too much weight on one part of the analysis and did not fully account for the functional experiments, the Danish comparison, and the combined ERAP2 evidence. They maintained that the ERAP2-linked variant remained their strongest selection candidate. So where does that leave us? With two separate propositions. First, ERAP2 variants genuinely influence immune function and are associated with infectious and inflammatory disease differences. That is well supported. Second, the Black Death rapidly drove one ERAP2 configuration higher in medieval Europeans. That proposition remains contested, and the distinction is essential. A gene can affect plague immunity without the Black Death having dramatically transformed its frequency. Even if the strongest ERAP2 claim is weakened, pandemics can still leave genetic legacies. Infectious diseases have been major forces in human evolution. Malaria, for example, influenced variants affecting red blood cells. Other pathogens shaped immune genes. A pandemic as severe as the Black Death almost certainly created powerful selection pressure. But its genetic effects may have been spread across many genes, with each contributing only a small survival advantage. Those effects may also have differed from region to region. A protective variant in London might not have been equally useful in Denmark, Italy, North Africa, or Central Asia. Different populations carried different genetic backgrounds, diets, climates, and disease burdens. They also experienced different patterns of exposure. The Black Death itself was not one uniform event. People were exposed through different routes and developed bubonic, septicemic, or pneumonic forms. Local mortality varied, and plague outbreaks returned for centuries. Migration after the pandemic reshaped populations further. Survivors moved into abandoned towns, and workers traveled toward labor shortages. Communities mixed, expanded, and formed new family lines.
All of these processes can alter gene frequencies without a clean before and after signature. The true evolutionary legacy may be more complicated than one mutation rising to dominance.
It may resemble a faint mosaic scattered across multiple immune pathways.
Some pieces may be detectable, others may be hidden by migration, drift, and later epidemics. Some apparent signals may disappear as larger, better-dated genome collections emerge.
That is not a failure of science. A dramatic claim is proposed, tested, challenged, and improved.
The story becomes less sensational but more accurate.
Research published in 2026 shows how quickly plague genetics is still changing our view of the past. Ancient bacterial DNA was recovered from hunter-gatherer burials near Lake Baikal. The evidence revealed deadly plague outbreaks about 5,500 years ago, thousands of years before the medieval Black Death. The finding concerns ancient plague bacteria, not a new human mutation caused by the Black Death. But it expands the evolutionary timeline. Human populations confronted Yersinia pestis long before the crowded cities of medieval Europe. Plague may therefore have pressured human immunity across prehistoric and historical periods, not only the 14th century. Some immune variants associated with plague resistance may have been shaped by infections thousands of years before the Black Death.
The 14th century pandemic may have reinforced older patterns rather than creating them from nothing.
Future studies will need more ancient individuals and tighter archaeological dating. They will also need much broader geographic representation. Most existing evidence is heavily weighted toward European populations. Yet the second plague pandemic affected societies across a far wider region. Without samples from many populations and periods, scientists cannot describe a universal human genetic response. So, does the Black Death still live inside you? Not literally. The plague bacterium is not hiding in your DNA. No scientist can inspect one immune variant and declare that your ancestors survived the Black Death.
But the larger idea remains plausible. Catastrophic epidemics can influence which variants pass into future generations.
RAP2 is a genuine immune gene whose variants affect how cells process immune signals. Those variants are associated with different patterns of infectious and inflammatory disease. A major ancient DNA study proposed that one ERAP2 configuration was strongly favored during the Black Death.
Other scientists challenged whether the ancient samples actually demonstrate that evolutionary shift.
As of 2026, the responsible conclusion is not that the mystery has been solved.
Researchers have identified a compelling candidate and an equally important controversy.
Somewhere in our genomes may be the record of epidemics survived by people whose names have been lost. But reading that record requires caution. DNA is a damaged archive shaped by ancestry, migration, chance, and environment. The Black Death unquestionably transformed human history. Whether it transformed specific genome regions as dramatically remains open, and that uncertainty may be the most fascinating part. Do you think future ancient DNA discoveries will confirm that the Black Death reshaped immunity or reveal a more complicated story? Share your view in the comments. Subscribe for more documentaries about disease, genetics, and human survival. And watch the next episode to discover how plague began thousands of years before the Black Death.